Hello and welcome back. The topic for this video, we're now going to talk about how eukaryotic transcription is regulated. And we've got a lot of other options here to talk about and a lot of different levels where genes can be turned either on or off. Okay? First one that we'll talk a little bit about is going to be DNA packaging. Because eukaryotic cells, unlike prokaryotic cells, are going to package their DNA. All right? And with eukaryotic cells, really with gene regulation, all right, where this becomes important in eukaryotic cells, this is going to be what accounts for the different cell types that you're going to learn more about when you take A and P that make up the body, okay? Because gene regulation is part of the process known as differentiation, okay? And differentiation means that cells go from having everything to having subsets of genes expressed that are important for the particular function, okay? There are going to be genes that are going to be important in a liver cell in order for a liver cell to do its job. The genes that are needed for a liver cell to do its job are different than the genes in your kidney. So you're going to have a different set of genes that will be active in your kidney cells versus your liver cells, okay? And that'll be true of all the tissues that make up the body. You've got different sets of genes being expressed, okay? There are certain genes that are needed for muscle cells, all right? You need lots of actin, you need lots of myosin. Kidney cell, you're not going to need a whole lot of actin and myosin in the kidney because the kidney isn't responsible for movement. Kidney is responsible for filtration. So what'll happen with cells, okay, cells are going to turn on the genes they need to do their job and keep the genes off that are not necessary, okay? Because every single cell in your body has the same blueprint of DNA, but the cells that in a particular tissue only read part of that manual, okay? So the gene expression in eukaryotic cells is really what allows for differentiation to happen. That process of differentiation is essentially a way of specializing cell function, all right? And the ways that that can happen, because we're talking about such a large volume of information in each cell, we've got multiple ways to do that, okay? So DNA packaging is going to be one that we'll talk about, okay? DNA essentially is going to be wrapped in proteins. And when that DNA is wrapped up in proteins, transcription can't happen because you've got all those proteins tying things together, okay? We are also going to have sometimes tags that will be attached to DNA. These are going to be epigenetic factors. And I'm only going to give you one example of an epigenetic factor. There are more than just the one I'm giving you. But the process of methylation is one way for the eukaryotic cell to tag certain parts of the DNA, okay? That methylation essentially is the attachment of a methyl group onto the DNA itself. It's a way of tagging the DNA, and that then is interpreted by the cell as far as this gene gets transcribed, this gene doesn't. Okay, so it's a way for the cell to communicate that information. Okay, typically when you've got methylated DNA, it's going to be off. Unmethylated DNA, lacking those methyl groups, that would be the on position. Okay. Then we're going to have also ways to control transcription. And I talked a little bit about this when we talked about transcription. And that's going to be the microRNAs. Okay? MicroRNAs are going to be short sequences of RNA. And what they're going to do is they're going to bind DNA, they're going to bind other RNA products, and they're going to regulate genes that go to be transcribed, not transcribed, translated into proteins by binding to RNA or DNA. Okay? So microRNAs is another way that we can regulate genes. All right? You're also going to have, as far as gene regulation goes, and I talked about this also with a little bit during the transcription video, is the process of alternative splicing. So making different RNAs that have different functions. That's also a way of regulating how genes are expressed. All right? And then our last one is going to be a little more analogous to what we talked about with the lac operon. And we're going to have promoters, and in the case of eukaryotic cells, something that makes them different from prokaryotic cells, we are going to have, there we go, regions known as enhancers. Those enhancers make transcription more likely. And we're going to have a subset of special proteins that are going to be involved in getting transcription to happen. And those are going to be transcription factors and activators. And I'll talk about those a little bit, I can speak, I promise. We'll talk about that a little more specifically in the next video. I'll go into a little bit of detail on all of these. I'm not going to go into great gory detail on any of them. Your textbook doesn't go into great gory detail either. Just know we're kind of scratching the surface, and what I want you to take away out of this really are the differences between prokaryotic and eukaryotic transcription. Prokaryotic transcription, much, much more simple. Eukaryotic transcription gets much more complex, okay? So keep that in mind. If you have any questions, please let me know.